Hi, so my name is Theopi. I work at Brandeis as a staff scientist. And today I'm going to talk a little bit about my work with halophiles, which are the super salty organisms that live in very salty lakes. So my lab life started in Florianópolis in Santa Catarina in Brazil, where I grew up. It's a beautiful island. It has these beautiful sunsets. And over there, what I did was I worked in a lab in the Universidade Federal of Santa Catarina, where I used to work with parasites that the larvae of these parasites can lodge themselves in your brain. And it's very hard to detect them unless you do a very expensive MRI scan. And for Brazilian public health, it would be very interesting to have a cheaper test to detect that disease, or at least to screen for that disease. And what I did in lab was I helped develop a blood test, so a much cheaper test to diagnose this disease. From there, I went to the largest city in South America, São Paulo, to continue with my grad studies. And in São Paulo, I worked at the Universidade de São Paulo, where I did my master's in molecular biology and biochemistry. So over there, what I did was I worked with Bacillus subtilis, which is a bacterium that lives in the soil. And I worked with the divisome, which is this division complex that forms in the middle of the cell and allows the cell to divide. And I tried to understand how some proteins are produced and degraded when forming the divisome. So that was my grad school work. Currently, I work with haloarchaea. So haloarchaea are organisms that live in very, very high salt concentrations. And one of the most interesting things about them is they have a myriad of shapes. So just so you have an idea, in this picture, you can see a bunch of the different cell shapes of haloarchaea. All of these pictures were either taken by us or by very close collaborators. So you can see that there's so many different shapes. They're in even triangular cells. They're square cells. There's everything that you can imagine. And what makes archaea so awesome is not all, but many archaea are extremophiles. So they live in very high temperatures. They live in very acidic conditions. So over here, you can see what an acid does to an orange. And they can live in a pH close to 0. So some species of archaea have been found in living around like very high heavy metal concentrations, very high temperatures, very acidic situations, very basic, super alkali ground. And some of them can live inside of brines trapped in salt crystals. So that pink Himalayan salt, the fancy one that you buy super expensive in the supermarket, it is pink because of archaea. And how salty are we talking about? So when we talk about sodium chloride concentration, our blood has 0.9% sodium chloride, seawater has 3.5% sodium chloride, and Halophorus volcanii, which is the organism that I work with, lives in about 18% sodium chloride. So that is very, very salty. And the way that they adapt to this is they actually pump salt into the cell. So it's not like they're just living in a very high salt concentration and pumping a bunch of salt out. They're actually living in homeostasis with the environment. And if you throw them in water, they die. So my main project is actually studying the biological aspects of the clusters that Olivia presented a couple of weeks ago. So I'm not really going to get into that. The thing that I'm going to talk to you about is my side project. So my side project, if you know, you probably have heard about the discovery of antibiotics. So very briefly, Alexander Fleming found antibiotics when he had a plate of Streptococcus. This was 1928. And he had a plate of Streptococcus in which a bunch of fungi started to grow. And what he observed was death of bacteria around the area where the fungi was growing. And he was like, that's very interesting. What is happening? And then he found that the fungi were actually producing a substance that could kill the bacteria, which is called penicillin, and we use it nowadays. And people have been discovering antibiotics by pitting bacteria and fungi against each other for years. This has been done for years. But guess what hasn't been done? What hasn't been done is getting a bunch of archaea and pitting them against each other. And at my lab, we have 90 different species of haloarchaea from all over the world. This is just a map, so you have an idea of where we find the archaea that we have. And my coworker and I just started as a fun project pitting archaea against each other. So when you grow them individually on a plate, they look like this. When you grow them side by side, most of the time, they look like this. We call this project Poke Battles because it is basically literally just pitting archaea against each other. But interestingly, we have already found some archaea that seem to kill other archaea in their vicinity. And now we are investing some of our, side project time to investigate these species and try to understand what's happening over there and what substances they're producing. So this has been done for one species of archaeon. There is one published halocin, which is the substance that kills other archaea, but it has not been done in this scale. And this is one of our side projects in the lab. So we're super excited. You can reach out to me. This is my email. And I thank you so much for letting me be here.